Okay, nucleic acids. They're called nucleic because you find them in the nucleus. They're associated with the activity of the nucleus. And they are all the type of chemical that we've met already, which is that they are polymers. But there are two different ones that we're going to meet. We're going to meet DNA and we're going to meet RNA. And their names are almost the same, but DNA is deoxyribonucleic acid. Whereas RNA is just ribonucleic acid. And the ribo in both of those comes from ribose, which is that 5-carbon sugar that we met in the carbohydrates. So I've got deoxyribonucleic acid and ribonucleic acid. Now, DNA is the one that we're going to concentrate on, and then RNA we'll sort of compare against it. And then when we look at protein synthesis, they both have quite specific roles. They are both what we would describe as polymers, chemically, which means that they are made of repeating units. So you've got individual units joined together in chains here. And the monomer units are called nucleotides. And the DNA that you might find in the nucleus of a cell would be, and I can't remember the actual number, but millions of nucleotides in length, but chopped up into sections that we call chromosomes. Chromosomes are made of DNA. The DNA is a polymer. Now, the way it's arranged in the nucleus is something that we will come on to later, but I will put it in here. DNA is not just randomly sort of, tangled up in the nucleus. It is actually organized, because if you think of how you would use information from a great big sort of strand of a chemical. If you take a human cell, it's got about 2 meters of DNA, right? 2 meters crammed into a nucleus, which is perhaps about, say 10 micrometers across at most. So, how are you going to get 2 meters of material into that and use it? You can't just have it stuffed in randomly like the cupboard under the stairs. Go on, please tell me everybody's got a cupboard in the stairs where everything's stuffed in randomly. Because it's a lot easier to find things if it's a bit organized. And, so you might put things into boxes or onto shelves or into cupboards or, that sort of thing. And it's the same with DNA. DNA, when it's being organized, is wrapped around proteins called histones. Now, we used to think that the DNA that we've got was it, as far as our genetics is concerned, and it's seriously not. That, that 2 meters of DNA that we've got, actually only about 4 centimeters of it is genes, right, giving the code for the proteins. So what on earth is the other 1 meter 96? We used to call it junk DNA. We've sort of moved on to calling it non-coding DNA, but in fact we actually now know that a lot of that does other jobs, and it helps to control the way that genes are expressed. So we're finding out a lot more about DNA all the time, and I think, if I was going to do another degree now, I can't do another one, haven't got the time, I would probably do genetics, I think, because it is the area of biology that's moving forward all the time. We're finding out more and more and more. So what we have in a cell, DNA plus, and we did this right at the beginning when we were doing cell structure, these proteins called histones is the substance that we see in cells between divisions, and it's called chromatin. So when you looked down the microscope at your own cheek cells and you looked at the plant cells and you could see a nucleus and it might look like a little splodgy thing, those splodges are this stuff called chromatin. So it's not just DNA. Now, we also used to think that we inherit DNA from our parents and that's it. What we get is what our parents have given us. To a certain extent, that is true. But it was always thought that you couldn't change your DNA. You can. Habits, particularly, things that happen during our lifetimes can actually change our DNA. So you don't change the code exactly. The code can change if you have a mutation, when the DNA gets copied and that sort of thing. You do get changes. We'll sort of come back to that idea. But you can get changes are possible in the DNA code, or the sequence, I should say, and that's what we'd call mutations. But it's also possible to get changes that aren't changes in the code, that might come from, for example, people's nutritional status, the things that people eat. So dietary influences can actually change the DNA. Other things would be things like smoking, for example, can actually change your DNA. And those changes can then be passed on to your offspring. But it's not changes in the code. The other changes are described as being epigenetic, which means around the genes. And in the next unit, and I'll keep referring to the final unit because the inheritance side of it overlaps with this a lot. But in that one, I will talk about epigenetic effects and the sort of changes that can happen to the DNA that will switch genes on and off, depending on things that happen to the DNA, but also to the histones, strangely enough. Other changes are described as being epigenetic. Well, not epigenetic. Epigenetic. And these affect the DNA and the histones. So during our lifetimes, things that we do could actually change our genetic information, and that will be passed on to the next generation. So it's it's a bit of a wake-up call actually, because we never thought that could happen. But it's a whole area of biology now. I've started a list on this flip chart of potential presentation ideas, and these are sort of coming up as I was talking last week, and I put epigenetics on that list because we will meet things in this unit that might be interesting to people. And epigenetics is something I've had students choose that for the last 2 years as a presentation idea. The other item on that list says mitochondrial DNA / 3-parent IVF, which I think I mentioned that when we were doing mitochondria anyway. But of course now, because we're looking at the DNA side of it, of course, that's why people might need 3-parent IVF. And of course that's now been approved in this country and I think has been used as well. Anyway, if we go back to the sort of chemistry side of this now, we need to start looking at what these nucleotides are, and that's really where the notes go in your handouts. And we're going to look at the overall structure of a nucleotide and then how you join them together to make a nucleic acid. And then we can move on to working out how you can use that sequence of chemicals to actually build a protein. And it's really important to have in your mind the difference between a nucleic acid and a protein. It's very easy to confuse the idea of nucleotides being the sequence in DNA and amino acids being the sequence in a protein. All right? But the sequence in the DNA is decoded to give you the sequence in a protein. They are not the same thing. All right. Okay, a nucleotide has got 3 components. So this is a bit that needs a bit of extra information. But in your handouts, you'll find that you've got an introduction to all of this. There is there's half a side that gives you a bit of the background, how the evidence came about for DNA as being genetic material. and a lot of individual people's work was really sort of groundbreaking on that. And then it gets into the bit on nucleic acids, describes them as informational molecules. You might think, is that a word? Yes, informational molecules. So they are there just to carry information. And we've now got, down at the bottom there, a bit of information about the component parts. So it tells you there, the sugar is either ribose or deoxyribose. Then there are 5 bases, and there are also phosphates in there. So I'm going to list those. But over the page, you have some diagrams that you might think, I have no idea what that means, but I will explain a little bit. If we'd done the organic chemistry bit, it would be a lot easier, but I will explain through what those mean. So a nucleotide has got 3 bits to it. It's got, first of all, it doesn't matter which of the nucleic acids, you have a phosphate group. Secondly, you have a pentose sugar. All right, I shouldn't need to say sugar actually, but I will. A pentose sugar, which means it's got 5 carbons in it. We have 2 options there. It's deoxyribose in DNA, and it's ribose in RNA. And when you look at models or diagrams of nucleic acids, those are usually drawn as pentagons for obvious reasons, because they've got 5 carbons in them. So if you look at the diagram in the handouts there, you've got ribose is up at the top, the topmost diagram. But interestingly, although it's a pentagon, that pentagon isn't made of the 5 carbons, is it? You can see the top point of the pentagon is actually an oxygen. Okay. Now, the carbons in that molecule, and it doesn't matter whether it's ribose or deoxyribose, the only difference you can see down at the bottom corner of it there, it says this oxygen removed in deoxyribose. But we have to number the carbon atoms. Now, we did this when we were doing the carbohydrates. We numbered the carbon atoms in the glucose molecules so that we knew we would join them together from a carbon 1 and a carbon 4, and the difference between alpha and beta glucose was how the groups are arranged on carbon 1. So we just number the atoms. There are rules for this. Doesn't matter particularly at the moment, but the numbering on this is critical. That carbon number 1 is over on the right-hand side of the diagram there, the sticky out point. Number 2 at the bottom right, number 3 at the bottom left, number 4, the sort of top left, and then number 5 is actually off on a branch on its own. Now, those numbers are going to be important. All right, so we've got, just to give you a very outline version of... No, because if you looked at it from the other side, it wouldn't be, would it? But there are rules for numbering that are based on which is the most important group in the molecule, and in this case it would be the one that the oxygen is actually attached to if you have it in a straight form, and that would be carbon 1. So there is a rule system for doing it, but we've got carbons 1, 2, 3, 4 and 5. Now, if you look further down, you'll find that those get changed a little bit. All right, those are just numbering the carbons in there. There's a reason why you need to know this when we start joining them all together. But this will become clear. The other thing that we have in a nucleotide is a thing that is vaguely called a nitrogenous base. There are 2 different types, and there are 5 different versions. In DNA, the bases that you can have are called adenine, thymine, cytosine, and guanine. And I wouldn't normally write them with capital letters, but we usually refer to them by their initial letters. And the DNA code is usually sort of described just in sequences of A, T, G, and C. All right, so that's the DNA option. With RNA, we don't have thymine. We have a very, very similar looking compound which has a very different name. We do have adenine, but instead of thymine, we have one called uracil. Then we have cytosine and guanine making up the numbers. That means there are 5 different bases: adenine, thymine, cytosine, guanine, and uracil. But again, you know, we refer to those by... I've given them capital letters because we would normally refer to them by their initials. So what I was then going to do is just underline how we would normally refer to them. There are 5 bases here, yeah. Adenine? No, there's 5. Adenine, thymine, cytosine, guanine, and uracil. There are 5 different bases, all right? So yeah, 4 in each one, but there are 5 different ones. Thymine and uracil, very, very similar chemically. Now, some of these are made of a single ring, and some of them are made of 2 rings. And you'll see those on your diagram sheet. The rings, yeah? If you look just under that 5-carbon sugar, you'll see 2 rings, yeah? All right, or 2 diagrams underneath it here. It's really important that some are made of 2 and some are made of 1. Because they're going to have to pair up in DNA. You know about DNA being double-stranded, don't you? This idea of the double helix. All right, I'll use our very simple model. All right, now what you have is the backbone of this molecule, and on this one it's yellow and purple, forming the sugars and phosphates down the sides, and the bases are paired up across the middle between the 2 strands. And you can see we've got dark blue pairing with light blue, and we've got dark orange pairing with light orange. But the light blue and the dark orange are bigger than the dark blue and the light orange. You cannot pair up 2 big ones, because that would push the 2 strands apart. And you cannot pair up 2 small ones, because they either wouldn't meet or it would pull the sides in. So you always have to pair a big one with a small one. The big one is made of 2 rings. So on your diagrams there, you will see the left-hand base, it says purine, 2 rings. And the right-hand diagram says pyrimidine, 1 ring. Okay. Now, it is important in terms of the pairing, but it works like this. The pyrimidines, 1 ring, and it's cytosine, uracil, and thymine fall into that category. The purines are the others. They're made of 2 rings. That just leaves us with, what does it leave? A, and that's it. A and G. Now, how on earth would you remember them? Well, you don't have to, really, because you're going to be able to take notes into the exam anyway. But I think that's not something I'm, you know, because... Well, yeah, you're taking 2 sheets in, that's all. You'll produce 2 sheets of notes. You can take 2 sheets of A4 in with as much on as you like, and it's, you know, it's up to you what you put on there. You won't actually need this, but a few years ago I was challenged to come up with a way of remembering this, and all I came up with, but it works for me and it sticks in my mind now, I don't have to make an effort to remember which ones are which, is give me a ring, all right, a ring, and I will see you for T at the pyramids. All right, not particularly sort of, you know, accurate in terms of what's that, but that is now stuck in my mind, so I don't bother trying to remember it any other way. So yeah, give me a ring and I'll see you for tea at the pyramids. And the other ones are the purines with 2 rings, so it's got to be A and G. But you won't have to remember that. It's just another, you know, just an idea. When you're trying to remember things, you know, you. But 1 ring, yeah, the cut, but you've got to get pyramids in there as well somehow. So yeah, I mean, you know, mnemonics and memory tricks and things like that are always going to be useful. So yeah, find your own. Now, what we've got to do is to stick together a phosphate, a sugar and a base to make a nucleotide, and you'll see that happening at the bottom of that sheet. The reactions that go on between the component parts are not important, but you can see a very similar theme to what we've already had with water molecules being lost, so that you've got H2Os coming out. So we had that when we were doing condensation reactions between carbohydrate molecules and monosaccharides. We did it with the fatty acids and the glycerol. We did it with the amino acids. We're always losing water. So it's the same sort of reaction again. If we put together the sugar, the phosphate and the base, we get a nucleotide. And there's a lovely little abbreviated version at the bottom there that works really nicely, so that you get your sugar is just drawn as a pentagon, with the phosphate group sticking off like it's holding a little balloon, and then from the side there you have the base. All right, so the base, the sugar and the phosphate, that gives us our unit for DNA or RNA. Now the next thing is to start joining these together, because one on its own is no good. We need millions of these to give us the DNA code. And so we start joining them together. If that is a nucleotide, then we could join 2 together and get a dinucleotide. Join 3 and have a trinucleotide. But what we tend to do is join many together and get a polynucleotide chain. Okay. So that is the nucleotide. If you get many of them joining together, you get the polynucleotide. And that really is what RNA and DNA are. Right? They are just polynucleotide chains. There is a difference, obviously. DNA and RNA are not the same thing. But we get a rather strange sort of effect when we start joining them together in DNA, because DNA is double-stranded. RNA is single-stranded, but sometimes folded. So I'm going to take you through the 2 different nucleic acids and flag up sort of some of the points about them. So, RNA, ribonucleic acid, and RNA is a carrier molecule in the protein synthesis process. We'll come across a lot of RNA, but it's not something that you sort of, you know, it's not part of general conversation, whereas DNA might be a sort of everyday term. Might be. DNA then, the thing about DNA is it's double-stranded. And it forms this helix. Oh, we keep meeting helices all over the place. We've had the alpha helix in secondary protein structure. We had an alpha helix in amylose, in starch. But this double-stranded helix, often just called the double helix, it's formed from two polynucleotide chains, and they are described as being anti-parallel. All right, now anti-parallel sounds a little odd, and I need to explain what it means. It will be important, not necessarily in this unit, but it definitely will in the next one. When we've got to copy the DNA before a cell divides, it matters that the DNA strands are anti-parallel. So the two strands they're described as being anti-parallel. Run out of L's. Now, this comes from the way that the bases will interact between the two strands. But what you get, if you continue looking at the diagrams in your handouts, at top of the third side, you can see it's saying nucleotides then join by condensation to form dinucleotides and polynucleotides. So you've got two joined there. And down at the bottom of that page, you've got a diagram of a polynucleotide. But as we go over the page, I haven't included a diagram actually until we get to the one which is on the sort of fifth side here. But I haven't included one that shows you this idea of the anti-parallel arrangement. So I'm going to have to try and draw this. Now, this is quite awkward, but we get, if we go, all right, I'm only going to do a bit of it because I get easily confused. All right, if we're joining together our sugar phosphate backbone, we would call it. Yeah, houses with little balloons. Okay, we would be going, I've run out of space, I'll do. And they would have bases sticking out. Let's say it's a single ring one there, and a double ring one there, and another double ring one there. It doesn't matter. All right, if that's one side, then the other side has to have bases which fill the space. So this one would have to have a double ring one. This would have a single ring one. This would have a single ring one. All right, so far so good. But to join these up, you do need upside down houses. Absolutely right. Yes. So I've now got to try and turn these the other way up and draw them with the point going downwards. Okay, so it goes like that. Actually, if I draw those in first, I do find this really hard to draw. Okay, we've got those in. And then we need, the phosphates. They're not quite lining up properly, I don't think, as I've got them. I've actually joined it to the wrong bit, sorry. Yeah, it goes to the bottom, not to the point. So let's just try that again. This is what I mean about it being so tricky. So it comes off there and joins to there. That's it. Comes off there and joins to there. Comes off there, and never mind what happens further down. That's the best I can do with that. Now, we've got the sugars, obviously then running anti-parallel. Yeah? But this gives us a way of identifying which, we can, if we look at the numbers of the carbons in the sugars, we can describe which way they're going. Now, the thing that I missed off actually at the bottom of the second side, when you had the diagram that joined together the component parts of a nucleotide, there is a little note down at the bottom that says formation of a nucleotide. Carbon atoms of ribose are, by convention, numbered 1 to 5, but with a little dash. Okay, right at the bottom of that page. So it looks like 1 dash to 5 dash. We actually pronounce that 1 prime to 5 prime. Right down at the bottom of the page where the nucleotides form. That side there. The bottom of the first diagram side. Right down the bottom, yeah? The thing is, there's an awful lot of carbons in those bases as well. They've got to have numbers. So if you can't number for the whole lot. So because we've got two components, the carbons in the bases just get the ordinary numbers, but the carbons in the sugars, once they're part of a nucleotide, we don't refer to them as I'd got them on the previous slide. Just let me go back and remind you what we've got. Here, we called them 1, 2, 3, 4, and 5. But when we attach something else to that that's also got its own carbons, we've got to distinguish between them. That's an oxygen atom at the top. So yeah, the 5 carbons are not forming the pentagon. Right? The pentagon's 4 carbons and an oxygen. A bit like with glucose, we had 5 carbons and an oxygen. So the 5 carbons there, if it was just ribose on its own, we would call them carbons 1, 2, 3, 4, and 5. But once we've attached a nitrogenous base, which, if you look at the diagrams of those, they're full of carbon as well, we have to distinguish between the two. Now, if we try and identify where the carbons are on here, the two really important carbons are 3 and 5. They're the ones that join up. They're the ones that form the backbone. We have number 3 here and number 5 there. But, of course, we've got to be precise. We've got to say 3' and 5', because these have got carbons of their own as well. So we'd have another 5 there and a 3 there, a 5 there and a 3 there. It's not a dot, it's a little dash. Yeah? Right, so we just pronounce it prime. But of course over the other side, it's the other way up, isn't it? So we've got a 3' there and a 5' there. 3 there, 5 there. 3 there, 5 there. And this is how we refer to the two strands running anti-parallel.
What does the little one mean?
That's what we're saying, prime. That means it's in the sugar, not in the bases. Okay? We have to number the carbons in the sugar, and we have to number the carbons in the bases. So that's what that little thing means at the bottom of that page that you just looked at, where it says, by convention, we number the carbon atoms in the ribose 1' to 5'.
And it primes in the sugar.
Prime is that's the little dash that just separates it from all the carbons that are in here and in here. They've got their own numbers. All right.
Prime is the carbon in the sugar.
Yeah, 1' to 5' are the carbons that we find in the sugars, as opposed to the carbons that we find in the bases. So when we talk about this, we can talk about the 5' end and the 3' end. And for the left-hand strand here, this would be our 3' end, whereas up here we've got our 5' end. For the other strand, the right-hand strand, of course, we've got the 3' end up here and the 5' end down here. And all that is saying is that the sugars are the other way up. Well, the whole nucleotide, of course, is the other way up. Now, the point where this is going to become really important is when cells divide, which is in the next unit, we have to copy all of the DNA and give a set to each of the new cells, don't we? Otherwise, we just keep halving it until there wasn't any left. So you've got, let's say 1 skin cell, and you want to make some new skin cells. So you want to divide 1 into 2. Well, you've got to copy all the DNA and then put a complete set in each new cell. So how do you make new DNA? It's going to be a matter of separating these two strands and then building new ones against the originals. And there are enzymes that do this, and the enzyme works better going, and I can't remember which way it is, it's either 3' to 5' or 5' to 3', and it causes some interesting effects, shall we say. Well, what it has to do, if it goes well, let's say it goes well 3 to 5. I just can't remember at the moment. It's not sort of in my head at the moment. It could go like that up that side really easily. How it'd have to do it on the other side would be to go like that, and then like that, and then like that, and join up all the bits. Yeah.
But we don't know that.
Not till the next unit, so I'm going to take that off there because that, yeah. But you know, when I refer to 3' and 5' in the next unit, that would be because it's come out of this one. The other option would be for me to put these 2 units together and make them a great big 9 credit unit, but yeah, that's not ideal. The strands then are going in opposite directions, but clearly there is something happening between them. We've got what we call base pairing. And when we talk about DNA, we very often see it written as so many base pairs or so many kilobase pairs, KBP. And a base pair really just means one matched with the other. So it's a single piece of the DNA where one nucleotide pairs with the other. But these in the middle between the 2 strands are the base pairs. Okay. Now there is a rule for how they pair up. And you will find that as you go a little bit further through. So yeah, the sugar and phosphate groups, you can see at the top of the page there, you've got a short paragraph, the sugar and phosphate groups forming this backbone. But the bases are the actual DNA code, so it's those that the sequence is important. And the way that they pair up is a very definite, single ring with double ring, so purine against pyrimidine. You can't pair them any other way. And they interact with each other, forming hydrogen bonds. Now, we did briefly mention hydrogen bonds in chemistry, which is where you have these weak interactions. It's a sort of bonding that you get between individual water molecules. So if you've started your biology assignment and not the biology assignment at all, it's your chemistry assignment, isn't it? If you started working on the section on water, one of the important things about water is it forms hydrogen bonds, right? But the base pairing is an absolute fixed arrangement where you will see just under that little paragraph, you've got pairing between adenine and thymine and pairing between guanine and cytosine. All right, now adenine is a 2-ring, purine. Thymine is a single ring, pyrimidine. Whereas with the other base pairing, guanine is your 2-ring, purine, and cytosine is the single ring, pyrimidine. So it's 1 to 2 each time. But they form different numbers of hydrogen bonds. Now, if you look at those diagrams, the dotted lines represent hydrogen bonds. How many hydrogen bonds do you get between adenine and thymine? 2. And how many between guanine and cytosine? Yep, got them. So adenine and thymine, I'm just going to put them with 2 lines like that. It's not a double bond or anything, just 2 hydrogen bonds. Cytosine to guanine is how they pair. Now that is something that you'll need to remember, A to T and G to C, but it's quite easy because how are you going to remember how they pair? Just look at them, look at the letters, how are they pairing? Can you see anything common between the way I've drawn an A and a T and between the drawing of a C and a G? No? Yeah? What can you see? You're missing the really blatantly... Yes, they're both curved. The A and T are both straight. Yeah? It is that simple. That's how you remember it. That's all you... Oh, yeah, I just wouldn't go any further, really. But, yeah, A and T are straight lines, C and G are rounded. It is as simple as... That's how to remember it. That's how everybody remembers them. Okay? So if I give you a sequence of bases on one strand of DNA, you can give me the code on the other strand. Now the 2 strands, one of them forms a template for making proteins. The other one is a non-template strand. but rather strangely called the coding strand. Yeah, and for I think almost my entire teaching life, I've actually been using the wrong word for that, and somebody discovered it last year, and it was an incredible professional shock, actually, that I'd been using the wrong word. All my processes were right, and my descriptions were right. I just used the word the wrong way round. So I try and avoid the word coding, because it makes a lot more sense to say template and non-template strands. So the way it's arranged, if I leave myself a bit of space on the left there, if this is my template strand, let's say, oh, it doesn't matter which, actually. Let's have them both in. Let's do the template at the top. All right, so that's the template strand, sometimes called non-coding, which is weird to me. Yeah, it just doesn't work for me, so I've got to stop thinking of it that way. But that's the code that is read during protein synthesis. If we've got code on that, we're going to have base paired matching code on on the other strand. But this one we call the non-template strand. But that is the one which is known as the coding strand. I suppose it gives the code for the other one, which is weird. And if I've got a code on here, let's say it goes A, T, C, G, G, C, T, T, G, A. You can tell me what's on the other strand. If that is how it goes, so the code that we'll get on the non-template strand reads. Yep, yep, yep. Yep, 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 yes, yeah, yeah. I realise that will sound very strange and it's recording. Me going, yeah. All right, so that's how it works. It is literally that sort of pairing. Okay. There are only those 4 letters in DNA. If we were making RNA, we make RNA from the DNA strand, from the template strand. It'll be the same sort of process, except we don't have any T's in that. We have U instead, so there's still only 4 letters. But we'll do that when we get to the protein synthesis bit. So that's DNA mostly. The only other thing really to say about it is that there are 2 types. No, that's not entirely true. But the types, the one that we're most familiar with, well, we've got nuclear DNA, and that's the one that carries our genetic code from generation to generation of cells and generation to generation of individuals. So there's nuclear DNA sitting in the nucleus. Where else do we find DNA? Think back to your, think of your very first assignment, mitochondria, yeah? And strangely enough, it's actually up on the board there, all right? So mitochondrial DNA is another one altogether, because of course we only inherit that from mothers, that's right, yeah. So nuclear DNA, we actually inherit half from our fathers and half from our mothers, so we get a bit of a mix and match there. But mitochondrial DNA is all from our mothers, and mitochondrial DNA doesn't determine any of our physical characteristics. It just determines the processing stuff inside mitochondria, produces the enzymes and things that mitochondria need. So there's a bit of an outcry in some areas about mitochondrial DNA in the relation to the 3-parent IVF. Oh, you know, if people have got 3 people's DNA, what will that mean for them? Well, it won't, because yeah, it's 2 parents and yeah, donated battery packs, if you like. So there are those. Now we could add, of course, in plants, they've got chloroplasts in their cells, which also have a bit of DNA in, but we won't add that here. And then you can artificially produce DNA as well. So in genetic engineering, there's production of artificial forms of DNA, but in our cells, we could assume that there are those. We might need some others as we go along, but yeah, the nuclear one is the one that we're going to concentrate on. RNA we are not going to say very much about at all. This is single-stranded, but it is sometimes folded. And remember, you know, it's got a different sugar in it. It's got slightly different bases, but still only 4. And the types that you get, well, there are more and more all the time that we didn't know about. But we have 3 that we're going to meet later on to do with protein synthesis. So we'll put those in first. We have one called messenger RNA, so that's usually just known as mRNA. And that one is basically a copy of the code that's used in protein synthesis. DNA is a bit like a reference library. You can't take it out of the nucleus. So you can go in there and have a look at it and copy something out and take the copy away, but you can't take the DNA out. So when we're making proteins, we have to go into the nucleus, copy out the recipe for a protein, and then take it out of the nucleus to use it. And that's what messenger RNA is. So more of that later. There's one called transfer RNA. Not surprisingly, that's a sort of carrier molecule. And it carries amino acids about, ready for protein synthesis. Then we have the other one that's most commonly quoted is ribosomal. Can you remember ribosomes vaguely from the first unit? Roughly are, and they carry out protein synthesis, yeah. So they're actually made partly of RNA. Then there are, and I'm going to put others with a question mark, because particularly when we get into the next unit, we're going to come across, it's something I've only just written into that unit. Some of this non-coding DNA that we've got codes for short pieces of RNA that control genes. And there's a whole area of gene control called RNA interference, where you get little RNA molecules going along and interfering with genes and stopping them being expressed. And so we're finding other sorts of RNA all the time, so you know, might come across those. Okay, that's all I'm going to put on there. Now, I'll take you quickly through the PowerPoint you haven't got, because I don't want to put too much into the handouts. You do have a sheet that compares DNA and RNA, but let's have a look. In here somewhere. Nucleic acid structure. Okay, that's my first slide. That was me just sort of playing around with animations to see if I could get them into PowerPoint, which you can, obviously. Did it with the heart as well, I seem to remember. One of the things that we now are going to start working towards in study skills is, oh yeah, that's due today, isn't it? Or sometime, is the final learning outcome in study skills, of course, is the presentation, which most people find, you know, quite a daunting prospect. But the first thing I'm going to do is to sort of go through the use of PowerPoint in presentations, and I will show you some bad ones as well as some good ones, and I'll show you some example ones as well. And I'll give you, well, I'll get you to do little practice ones as well. However, yeah, this was years ago, me just trying to find an animation that I could include in, and it worked quite nicely. Now, In terms of what DNA is on the larger scale, this isn't something that we've met yet, so it's a little bit difficult to talk about DNA without mentioning chromosomes. But if you take a typical human body cell, let's say a skin cell or a white blood cell, why wouldn't we take a red blood cell? No nucleus, exactly. So no DNA in a red blood cell, which is why they don't live very long. If you take a white blood cell or a skin cell or a liver cell or a kidney cell or something like that, it has a nucleus. And when that cell divides, the DNA in the nucleus organizes itself into chromosomes, which then organize themselves and divide up. But within a chromosome, which is made entirely of DNA, you have sections called genes, and it's the gene which is the code for making a protein. All right, so that's the bit that we're going to come back to. So it will, this will become clearer. If you looked at it, in terms of the nucleus, we've got on there a nucleus with chromosomes inside it, and if you unwind the chromosome, you get the DNA, and a section of that DNA is a gene. Looking at it on another scale entirely, if a chromosome at the bottom being gradually unwound into DNA, or you could take the DNA at the top being gradually wound up into a chromosome. But in order to fit 2 meters of DNA into a nucleus, you've got to wind it up and organize it. And it's a bit like, if I was saying to the other group, the old-fashioned knitting wool used to come in what we described as skeins, which was just big long loops. And I can remember spending time as a child sitting with my hands in the wool while Mum just wound the wool into a ball to get it organised. I mean, you buy it ready wound now, don't you? But we always used to recycle wool from old knitted stuff as well, and you'd unravel a jumper and wind it up and knit it back together. And there's that idea of winding it up so it's organised, so that it doesn't tangle as you're using it. And that's what we've got with DNA, really. If you take the DNA, you wind it around these proteins, which are the histones, and then pack those up and fold them, and eventually you get a chromosome. I think there's another one of those. No, there isn't. Right, do those 2 names mean anything to anyone? Watson and Crick? No, they really, yeah. Watson and Crick, I can't describe how important their discovery was. They discovered the double helix. For a long time, people had known what DNA was made of, and they could extract it, and they knew it had got sugars and phosphates and bases, but they couldn't work out this double helix idea. And so Watson and Crick, they got all the credit for this. It's one of those iconic pictures in science. I don't suppose that was the moment they discovered it, but that was, pose by your model. And one of the most remarkable things in recent years is, I've forgotten which one of them it was. I think it was Francis Crick. Yes, Francis Crick. And this was from 4 years ago now, almost exactly 4 years, April the 9th, 2013. His son, I believe, sold a letter that his dad wrote to him when he discovered it. So in 1953, all right, he'd written this on March the 19th. Oh, it must almost be. 1953. And Francis Crick wrote this letter to his 12-year-old son explaining what he'd discovered. And Jim Watson and I have probably made the most important discovery. We've built a model for the structure of DNA. And he goes through and he's got this sugar phosphate backbone with the bases sticking off it. And this is just a letter to his son. And then, describing it like, and it's really not changed since then. So, going back to 1953, that's 5 years, even before I was born. And, just the base pairing is in there as well, A to T and T to A.
I love that.
I think that is that's why I've included it in this PowerPoint now, because I just think it's just no, exactly. Yeah, I think it's really important to involve young people in everything. They need to be understanding and not just reading rubbish. But the trouble is, of course, you know, in those days, I mean, this is how information was passed about anyway. You know, there was no texting, emailing, or anything like that. You know, it was all done by post or telephone. And yeah, I mean, it is quite readable, to be honest. I don't know how much it sold for in the end, but quite a lot. But I don't know, if I owned that, I wouldn't want to sell it. But I have a sneaking feeling it probably went into, you know, into a museum or something like that. But you know, I think that's it's a little rather sort of fantastic little snippet there. Anyway, yeah, so he came up, they between them came up with this idea then that you'd got these there's anti-parallel strands, so you can see sometimes easier to watch it with where the oxygen is, so oxygen down on that side but up on the right hand side, with your phosphates in between. This is a bit more filled out than the ones that we've looked at, and then the bases pairing across the middle, but not a lot of detail in it, and you find so many versions of it. The double helix is the whole molecule, all right? A helix is a spiral, and because there are two strands, it forms a double spiral, so that you get, you know, one on its own would be a single helix, but because there are two strands going round, it's a double helix, yeah? But to actually come up with the idea that it was that, obviously took quite a lot of work. So what did they do? RNA, it varies in shape, to be absolutely honest. It might be, it's not likely to be a helix, actually, despite that diagram. It can be straight, it can be folded. We'll come across different versions of RNA. No, it's a single strand. So the base pairing then is like that. So you've got your adenine to thymine, cytosine to guanine. You can see the hydrogen bonding a bit more clearly there. The hydrogen ending up with a slightly positive charge, and then oxygen or nitrogen providing a slightly more negative charge, pulling the electrons. Remember about electronegativity and all that. And then you've got all sorts of different models of it. That is a very complex one that you can hardly see what's going on there, except you can see the sugars the different way up quite neatly on the two sides. And then there's a very simple version, but I would say there are at least two things wrong with that. That's a third thing. Yes, there's no difference between the size of the A and the T, the G and the C.
The arrows be the other way round for the other side.
Yeah, we haven't got it going anti-parallel, have we? So we've got both of them going the same way up, the sugars and phosphates. And the other thing I would say is, where are the bases, the A, T, C and G, joined on? Well, somewhere sort of vaguely between sugar and phosphate. Well, they're not. They're joined to the sugar. So I put that one up as a bit of a warning. You know, let this be a warning to you. Not all diagrams are okay. You know, you need to be understanding what they're showing you, and that one is pretty rubbish, actually. Yet another model, so this idea of the double helix, you know, the double twisted arrangement, and lots more as well. And yeah, so you must, you know, wonder, well, you know, how on earth did they come across this idea? That, I don't know whether you can see that one, but it's just a stick model showing the double helix, but the reason I put that one in is because I also got one like that. Now, what does that show? What are you seeing? That's exactly right. You are seeing it from the end. You see the exact, yeah, you get that idea. So around, you get the outside bit. It's a, yeah, bird's eye view, end on, you know, looking down the helix. You can see you've got the, I can't see exactly what we've got, but well, I can see a little pentagon there, which is a sugar. So I'm assuming perhaps the reddy orange bits out here are phosphate. So we've got all those around the outside. And then in the middle, this sort of area is where you're getting the base pairing. So, yeah, it would look like, yeah, blue and a bit of white. And I'm not sure, it depends what they're modelling. I think the blue is the nitrogen. White will be hydrogen, a bit like the bead models, because there's a lot of red, which would be oxygen. And yeah, so I think that's done like that. But a better version of that to be seen on one of these boards is that. That's the sort of, you know, the simplified version of that, where you can see the sugars and phosphates round the outside, the little pentagons with their sticks, and then the bases pairing across the middle. Ah, good question. You should know about Rosalind Franklin. It's very important that you know about Rosalind Franklin because she was a scientist at the same time as Watson and Crick. That is Rosalind Franklin, yes. Yeah. So we're talking, sorry? No, Rosalind. Rosalind, yeah, Rosalind Franklin. And she was working as a scientist at the same time as Watson and Crick. But of course, we're talking about the early 1950s when female scientists didn't really have a great deal of respect, perhaps, and their work wasn't necessarily considered or credited or anything like that. And what she was doing was a process called X-ray crystallography, where you pass X-rays through crystals and you get these sort of maps that tell you where atom density is. So you can work out the structure of molecules by X-raying crystals. She did this on DNA, and she produced this image on the left. Now if we go back to that, you can see what she'd found, but she didn't realise that at the time. This image went to Watson and Crick, and it was from that that they proposed this double helix structure, and she got no credit at all. So she provided the evidence they needed to come up with their theory, but didn't get the credit. By all accounts, she was a bit of a bitch to work with, so yeah, people sort of weren't that keen anyway, but you know, it wouldn't happen now, I hope, would it? You know, although, you know, I think there is still quite a male bias, I must say, in science. But yeah, she produced that, and so I think it's always important, you know, to remind people. Well, it's not random thought necessarily. It's usually progression. You know, we're looking at this now. Let's try that. But yeah, I mean, scientific discovery is, you know, 99% of the time they're finding nothing at all, and they're just sitting and thinking about it, and there will be a lot of mistakes, and a lot of mistakes actually open up new discoveries. And yeah, so you know, it is random some of the time, but you know, you've got to know what to pursue really. But yeah, so credit to her really. Watson and Crick got Nobel Prizes for it, she didn't. So you know, there we go.
Did she even understand what she found?
She would not have known at the time what she was looking at, but because she was imaging DNA, and I don't know how it got to Watson and Crick, but she didn't have their knowledge of what they were doing, so you know, they put it all together. So yeah. So there we are. Yes, we need to remember Rosalind Franklin. Now RNA, again, it's drawn on here as having a helix shape, which is a little bit misleading, to be honest, and it's really, I think, just showing that it's a single strand, whereas the DNA is this double. You wouldn't probably get the same helix because the helix forms by interactions between the two strands sort of on the outer edges. So what you tend to get are shorter pieces of it for a start, and they form all sorts of odd shapes like that, for example. Transfer RNA, it's still single-stranded, but it's folded up. See, so that's not a helix at all. They often describe that as a cloverleaf shape. We're going to need that when we get onto protein synthesis. And there's a model of the same thing, which is hardly any use at all. Right, okay. All right, we will probably, I think we'll stop and take a break at that point. If anybody has got the urge to build a model, then you can do that, or you can just look at the one that's already done.